a very kind introduction, and I was more than happy to come and chat and accept. It was very, very kind of you to sort of invite me. As I said, I'll leave the chat open. So if anyone, particularly trainees, wanted to write their question in throughout the seminar rather than actually interject, I can address it immediately. But please also interject with any questions if there's something that doesn't make some sense as I try and rush through so many disparate topics today. We can address it straight away. And so what I really want to talk to you about today is sort of the title of the talk is a question that I've used for a number of years now, and I still don't actually know the answer to it. So I'm really sorry if you're coming here looking for answers. What I did want to do was just talk about a few exciting discoveries that we've had in the last few years. I will talk very slightly about some recent work from a postdoc in the lab, Philip Hazel, but I know as we mentioned, he'll be coming to talk to you more in depth about that. And then I want to spend a little bit of time just talking about the sort of implications of some of the discoveries that not only ourselves, but others in the field have made as well. So what do reactive astrocytes really do? We don't really know ultimately what they do, but we know some things that they can do some of the time. And so what I really want to start by doing is thanking all of the people that have actually done the work that I'll tell you about today, either from the lab or from collaborating labs. And really the vignettes that I'm going to focus on today is some work that was spearheaded by Kevin Gutenplan up here on the top right. He was a graduate student that I was extremely fortunate to work with in my time at Stanford. And he was sort of like an adjunct member of my lab after Ben Barris had passed away. And then also I'll touch on some work by Philip Hazel, as mentioned. He'll be talking to in a few weeks' time as well. And I will disclose I do have a financial interest in this company, Astronautics. So you are happy to take anything I say with a grain of salt. So this is sort of how we think about the brain. And Ruth will be happy we have a neuron here, which is really great. But also we're putting this astrocyte front and center. And as a pharmacologist and a cell biologist, really the way I and my lab thinks about the brain is how two cells might interact together, and what are those interactions normally during development, during normal physiology of the brain, and then also during adverse context. And by that I mean in sort of the context of injuries or insults, infections, and also chronic neurodegenerative diseases. And so for most of today, what I'm going to focus on is a single function. That is the whole idea that astrocytes provide trophic support to maintain neuronal function. And this could be not only molecules that they need to survive, but also reuptake of glutamate, production of synapses, things like this. And so normally, the sort of very simple idea of the brain to me is astrocytes support neurons, and neurons do their job, which is to make us live and laugh and live and breathe and do all those things. But there are adverse conditions where astrocytes can change these functions, and we like to then think about both passive and active ways in which this can change. So one thing that can happen is that neurons can die in the extreme condition, and a passive way that this neuron could die would be that these astrocytes lose their ability to provide support. Maybe they deliver less trophic molecules to these neurons, and regardless of the susceptibility of those neurons, with less trophic support, they would slowly starve and then die. And another form of passive cell death could be if there is a lack of uptake of glutamate, and they could lead to excitotoxic cell death of these neurons. We also like to talk a lot about this active gain of function or active cell death that's mediated by astrocytes. And this is something that we have spoken about a lot in the last few years. And we'll have a very exciting discovery published very shortly, where we've actually identified the toxic factor that these astrocytes do secrete to cause the cell death of the neurons. But I'm going to show you today how this gain of function, this neurotoxic function, is not passive at all. It actually requires some susceptibility on the neuronal side as well. So it's a very active mediated cell death where there is a combination of something happening in the astrocytic side and also something happening on the neuronal side. And then, of course, there is a sort of an opposite to this, and that is that in the context of these adverse conditions, these astrocytes could become extra protective, uber protective, and provide additional trophic support to these neurons to help them weather the storms, so to speak. The neurons are damaged, the neurons are under stress, and the astrocytes do their best to actually preserve those neurons. And one of the things that we're really interested in now is Why does sometimes these pathways get engaged, and why sometimes do these pathways get engaged? And trying to understand that sort of pull and tug between preserving neuronal health and function and removing neurons from the circuitry. So there are really 5 questions that everyone in my lab then addresses in trying to come to this understanding of what it is reactive astrocytes do. And if you're interested in reactive astrocytes, you first have to understand what a non-reactive astrocyte does, or what an astrocyte does in normal physiological conditions. And there's been decades of work talking about these exact functions, and so I won't beleaguer those too much today. But of course, we are mostly interested in how these functions change in the context of disease or pathology. And the dirty little secret from cell biology land is normally what we do is we put astrocytes in a dish and we give them an acute insult, and we try and make some inference about what that means in the context of chronic pathology. And so are we really looking at pathology, or are we looking at acute responses? And because normally we're looking at astrocytes under normal physiological conditions. But ultimately, what we're building a repertoire of being able to do in the lab is say, how does the response to this secondary insult, and let's just say that insult could be an inflammatory insult, how does that change normally in adulthood with altered physiology? That is, how is it different during development, or how is it different with aging? And also, how is that response different with respect to chronic pathology? Are there changes in disease-associated genes or mutations that could alter the way in which this astrocyte would respond to that secondary insult? And does that change the landscape about what we understand about what these reactive astrocytes are actually doing? Is something that looks detrimental under normal physiological conditions actually having a beneficial effect under chronic pathological conditions or at different stages of development? So this makes for a very complex little rubric to try and understand exactly how these interactions are changing. We then, of course, are really interested in how different heterogeneous populations or substates of these reactive astrocytes could be changing across different disease states. Does each disease have its own associated reactive astrocyte subtype, or are there multiple heterogeneous subtypes that have some homogeneity across different insults? And we'll talk about that briefly today. And although I'm only going to talk mostly about the heterogeneity at both the gene level, we're also really interested in how this heterogeneity occurs at the protein level, small molecule level, lipid level, and ultimately what we care about is the functional heterogeneity of these cells, because the functions, the interactions between these individual CNS cells, that is something we're interested in. And then the last 2 questions that the people in the lab are asking is, to address all of that, what tools do we have? And we're really lucky that we have an amazing array of tools at our disposal in the astrocyte field nowadays that we haven't really had in decades previous. But also always the question, what tools do we actually need? And I'm not going to talk a lot about this today, but we are in the process of developing lots of reactive astrocyte reporter mouse lines, as well as the ability to synthetically genetically engineer astrocytes so that we can look at specific functional outcomes of disease-associated point mutations. And I'm more than happy to talk about that offline if anyone is interested. So if we talk just very quickly, this is a very simple cartoon that was made by Rachel Kim, a graduate student in the lab. It was in a review earlier this year. But we like to sort of think about what are the functions that astrocytes do have, and if they were altered in any way, how would that possibly be mediating the functional deficits we see in a whole host of different neurodegenerative diseases? So we already talked about the fact that astrocytes provide trophic support. If they didn't do that, what would happen to the neurons? We already know that astrocytes are really important for the formation and modulation of synapses, like pruning of synapses and things like this. If they didn't do that, how would that incur sort of insult in the brain? And of course, something that we've had a lot of interest in over the years is how these astrocytes interact with immune cells, either in the central nervous system or in the periphery. So we like to ask questions, are these functions lost in disease? And of course, there's lots of instances where that can occur. But maybe the functions are not lost entirely. Maybe there is a possibility, as we have here in orange, that maybe these functions are still present. They're just not as effective as they once were. So maybe these astrocytes do provide some trophic support, just not the same amount that they provided before. So the neurons are still alive, they're just a little sick. And would that change the way that we're able to fire electrophysiological potentials, and how would that affect the network and function of the organism? And of course, maybe it's not that they completely don't recycle these neurotransmitters, but maybe they're just not effective at it. And how would that sort of constant increase in glutamate that's not cytotoxic, but a constant increase of glutamate affect the way these neurons are affected? And so we're interested in cells functions that are completely lost and functions that may be decreased, but then we're also interested in functions that maybe are not normal and physiological, but are originating only in the context of these reactive states. And the two that are most easy to describe is, one, these cells can re-enter the cell cycle, become highly proliferative, and produce a scar or a border that can corral necrotic injury or try and help mitigate the infiltration of peripheral immune cells. And this has been studied a lot in the context of stroke or acute injuries and axotomy. And of course, something that we've become somewhat infamous for is the idea that these astrocytes can gain this potent neurotoxic capacity, and trying to understand exactly what is the evolutionary reason for having the ability for a CNS supportive cell to then turn into a CNS destructive cell. So I always like to start this talk with, actually, I don't want to call this historical because it's only a decade old, but this really sort of pivotal piece of work that was done by Jennifer Zemanian when she was a postdoc at Stanford. And in this study, she very simply took an astrocyte reporter mouse, an LDH101 EGFP mouse, and she gave it a systemic LPS injection to model bacterial infection or inflammation, and she ligated the middle cerebral artery to model an ischemic stroke. And when the astrocytes were isolated from these reporter mice and microarrays were run, she showed that there was very different transcriptomic profiles between these two reactive substates. And of course, we know that within these substates, there would be a lot of heterogeneity, but this is one of the very first studies that actually describe these differences that seem to be dependent on the initiating insult. And one of the things that I find really compelling that Jennifer wrote in the abstract of this paper, which I think people often forget, is it's not that there are only two of these reactive subtypes. She actually very sort of poignantly suggests it raises a really important question: exactly how many of these different reactive astrocytes exist? Are they either an infinite number of them dependent on all different infinite numbers of insults, or is there some homogeneity here? And of course, the really exciting thing for me, I think, was it's not the transcriptomic heterogeneity that is really important. It's trying to then use that transcriptomic heterogeneity to model these cells to understand their function. And I think we're sort of reverting back to this transcriptomic heterogeneity a little bit now with single cell sequencing. It's very, I don't want to say it's easy, but it's easier to do transcriptomic studies than it is to do functional assays and develop those functional assays and look at that heterogeneity at the functional level. So taking this paper as a starting point, and also the methods that Lynette Fu had developed also at Stanford to enable astrocyte culture in serum-free media, which was really important. I decided to take this particular inflammatory reactive subtype and try and work out exactly what molecules are required to induce them. So this is a paper in one page here. Essentially, the take-home message was that we found these reactive astrocyte subtype that was induced by inflammation. We knew that they were being induced by cytokine release from microglia, specifically IL-1, TNF, and C1q. And we used this knowledge to be able to make an in vitro model to study their function, and also to modulate their reactivity in vivo, either using genetic knockout mice or neutralizing antibodies. And at the time, we could also actually use single cell qPCR, because single cell sequencing wasn't a very big endeavor at the time. But we found that we could actually label these neurotoxic or neuroinflammatory reactive astrocytes with complement component C3. And that seemed to be very specific to particularly this substate of cells. And we could use that to go into human postmortem tissue and at least see that there was a correlative piece of evidence that where there were neurons dying, these C3 positive cells were present. But of course, we were really interested in the function. And so we could use this in vitro model that we generated to actually look at what the functional interactional changes were with these cells, with neurons and oligodendrocytes, microglia, and all the other CNS cells. And we did a whole host of these functional studies, but sort of the main ones that have been validated by lots of other labs around the world is that these astrocytes, when they enter this reactive state, fail to promote the formation of both structural and functional synapses. And they also seem to decrease their phagocytic capacity, so they're no longer readily able to either prune synapses or remove any cytotoxic cellular debris that could be present, for instance, myelin debris, which could be really important in the context of diseases like MS. And we also noted that there was this one gain of function they had, and that is that these cells secrete a molecule or molecules that is potently toxic to both neurons, mature neurons, and mature oligodendrocytes, but not to any other cells of the central nervous system. And that includes precursors to neurons and oligodendrocyte precursors as well. So it's very, very specific to these two mature cell types. Now, the caveat with all of this is that we did all of this study in rodent cells, and it was always niggling in the back of our mind that maybe this was a rodent-specific phenomenon. And although we could correlatively show that these cells were present in human postmortem tissue, maybe the cells are actually responding very different in humans than they are in rats and mice. And so when I moved to NYU, I partnered with Valentina Fossati's group just across Midtown here at the New York Stem Cell Foundation, and did some work that we published last year in Neuron, which was spearheaded by Lily Babar, who was in Valentina's lab, and Indigo Rose in my lab here. And what we showed was that if we use human iPSC-derived astrocytes, also, and very importantly, also grown in serum-free conditions, we could recapitulate transcriptomically this phenotype of reactive astrocytes by adding IL-1, TNF, and C1q. And we then had a cell-based assay that importantly, we could show that we had the same loss of function, a failure to promote the formation of synapse formation and synapse function, a decrease in the capacity to phagocytose a whole host of different prey particles, and importantly for this particular subtype, these reactive astrocytes in human reactive astrocyte substate were able to kill neurons by release of a toxic factor. So this was a really important validation of the original work that showed that this was an evolutionarily conserved substate of reactive astrocytes that is induced by the same mediators from microglia and has the same functional deficits as well. So then we got really interested because we had a model where we could actually drive the formation of these cells, either in rodents or in humans. And we could do this using primary cells or stem cell derived cells, either embryonic stem cells from mice or iPSCs from humans. And we knew that they were potently able to kill neurons, but we were taken by the fact that whenever we saw them induced in the brain, and often when we use a systemic inflammatory insult, we would see them induced throughout the entirety of the brain, but we wouldn't always see neurons dying. So there was this real disconnect between presumably toxin secreting reactive astrocytes present, but also neurons not dying. And so we decided to dig into this a little bit further over a number of years. And one of the ways that we did this was some work that was spearheaded by Kevin Gutenplan. Like I said, he was a graduate student at Stanford and adjunct with us here at NYU. He's now at the Volum in Portland with Mark Freeman. But we had showed previously that when we induced an axotomy that we did by using an optic nerve crush retroorbitally in the optic nerve, we saw that that optic nerve crush mounted an inflammatory response, which mounted a reactive astrocyte response in the retina that was transcriptomically similar to what we saw with our neurotoxic reactive astrocytes. And we'd done a bunch of experiments to show if we modulated this reactive phenotype from occurring, we could actually stop the retinal ganglion cells from dying. And so I'm just going to show you 3 little vignettes of an experiment that were published last year in Cell Reports, really digging deeper into this particular phenomenon. So the first set of experiments that Kevin did is, this is actually all in wild-type mice to start with. And what we're looking at here is just some quantification of the viability of retinal ganglion cells in the vertical axis. And we start with this gray bar here, in which we show if in a wild-type mouse, we damage the optic nerve, the cell bodies of those axons, which are the retinal ganglion cells in the retina, actually degrade. And this is interesting because also just from the anatomy of the retinal ganglion cell layer is the only layer of the retina that contains astrocytes. So there's a time and a place coincident here with this particular function. So we already knew that this occurred, but the experiment that Kevin then did was he took conditioned media from these reactive astrocytes that was neurotoxic in a dish and was able to kill neurons in a dish, and he injected it directly into the vitreous of the eye. And the hypothesis he was testing here was, if I don't damage the neurons, but I just present them with the toxin, will that toxin be sufficient to cause the death of the neurons? And we actually thought that it would be, and indeed he was able to show that it wasn't. So we interpreted this by saying that the toxin alone is insufficient to kill these neurons. And the white bar here is actually just a control where we've taken physiologically normal or control astrocyte condition media, which is full of trophic factors, and injected into the eye. And of course, we didn't expect it to kill, and it did not. So in the second set of experiments, we took advantage of an old mouse model that we used to use, which is an incredibly dirty mouse model. It's actually a global knockout for the 3 microglia-derived factors that induce this neurotoxic phenotype. So IL-1 alpha, TNF, and C1q. And although we know that these factors are involved in a whole host of important physiological, developmental, and other functions to deal with neuronal plasticity and circuitry plasticity, it enabled us to very quickly study this particular functional difference here. And we've since produced a lot more specific conditional or astrocyte conditional knockouts of molecules required to produce this toxin. But at the time, we knew that if we block the formation of these reactive astrocytes, we wouldn't see the death of these retinal ganglion cells. The 2 control experiments in this instance is one showing that in the knockout mouse, there would actually be no death of neurons by adding the trophic factor supported astrocyte condition media or the toxic astrocyte condition media, just in case there was a neuronal phenotype. But then what Kevin decided to do was take this injured triple knockout mouse, so damage the optic nerve. There would be no production of the reactive astrocytes, and then he would artificially and exogenously apply that toxin. And when he did this, he could see that the actual neurons were then dying again. And so we interpreted this, that toxin alone is insufficient to kill the cells, injury alone is insufficient to kill the cells, but combined, if we have the toxin and some injury present, there would actually be susceptibility imparted on these neurons, and they would be killed by this astrocyte-derived toxin. And this was exciting. It wasn't what we expected, but it was very interesting. It sort of fit with the hypothesis that we're formulating as time went on. But really, the most exciting part of this whole study was actually this group of neurons here. And this group of neurons is here is the neurons that would have normally died, but we were able to keep alive by mitigating this neurotoxic reactive astrocyte phenotype. And from a translational perspective, these are the neurons that ultimately would be maintained if we were able to produce a therapeutic that stopped the formation of these reactive astrocytes. And so the question we wanted to ask was, are these neurons still functional? And if they are functional, is there a difference in the susceptibility or the window of that susceptibility? And so we actually decided to do this not using an acute injury. We decided to slow things down a little bit, and we actually moved to using the Bead occlusion model of glaucoma. I won't run through exactly how that model works, but again, if anyone's interested, I'm happy to talk through that offline, as developed by Dave Colkin's lab at Vanderbilt a number of years ago. But in this particular model, you inject these beads, you see an increase in intraocular pressure, and then a month or so later, you see concomitant death of the retinal ganglion cells. And so all the data I'm going to show here was generated by Drew Adler, an MSTP student here at NYU, who at the time was in my lab, and Ben Stafford, a research scientist at the Salk. And so the first question that Drew and Ben asked was, if we elevate the intraocular pressure, if we get a glaucomatous mouse and we stop the formation of these reactive astrocytes, does the structure of these neurons, does the morphology of these neurons change? And so here we have, these are all in knockout mice. None of these are done in wild type mice. But we can see if we look at a slice of the retina through all the layers and we actually difill these retinal ganglion cells, we can see in the control or the uninjected eye, the dendrites of these retinal ganglion cells sit in a very distinct layer in this retinal architecture. Whereas if we elevate the IOP, that is, we induce glaucoma, but these neurons are not dying because there are no reactive astrocytes present, they're actually now sending projections and these dendritic projections to different layers of the retina that they didn't normally. So there's already a difference in this Z projection in the way that these neurons are morphologically looking. If we look in different projections, we can see here is a dye-filled retinal ganglion cell. We can see in the uninjected eye, they have these beautiful large arborized domains. And again, if we look in this Y projection, we can see that they have a very distinct dendritic layering that they are attached to. But if we look in the retinas of these animals that have glaucoma induced, the domain structure of these neurons is perturbed considerably, but also most importantly, the dendritic arborizations are very different. So these neurons look different if we keep them alive when they would have normally died. But what we really were interested in whether or not they function differently. And our hypothesis was that they would actually be functioning aberrantly, and it's that aberrant functioning that was then sort of imparting some susceptibility, and these astrocytes were then killing those neurons to remove them from the network so that you could rewire due to plasticity and sort of preserve the entire network stability. And so what Ben did here was he individually patched onto these retinal ganglion cells, and we have here again just the knockout mice in the control and a bead injected or glaucoma mouse. And we can see in the control setting, and I won't go deep into electrophysiology here, but we can see that there is a large number of these individual events, as noted by the vertical lines here in this electrophysiological trace. But in the bead injected or glaucomatous eye, there is a decrease in the number of these events. But what we found quite surprising was, although there was a decrease in the number of these events, or the spike rate, each individual event itself was almost identical to the normal healthy eye. So the decay of these events, the latency of these events, or even those retinal ganglion cells' ability to respond to gradations of light moving in different directions was remarkably similar. And so we sort of took this to think that there is actually some preserved electrophysiological function, which we did not expect, that we can maintain by simply modulating the reactive state of these astrocytes in the context of this inflammation that is induced following glaucoma. And if anyone's interested, we have all of this data. Like I said, we published it last year in Cell Reports, but just to run through the model very quickly, we think that these reactive astrocytes kill neurons in a two-step model. First step is that they present the toxin to the neurons, but the neurons also have to be susceptible for that cell death to occur. And we did this by showing that if we inject just the toxin alone, but there's no injury or susceptibility to these neurons, they are actually preserved. We know that if we remove the reactive astrocytes, but then both damage mechanically and exogenously apply the toxin, we can see those neurons dying. And importantly, the next step was, if we keep those neurons alive, we can actually then see that they have a remarkably preserved electrophysiological function that we did not expect. And so we think that this is a valid avenue for future therapeutic intervention. So the next question, I'm going to jump models here a little bit, because one of the things we've always tried to do is just increase the time that we're enabling these astrocytes to have an effect. So we started with an acute injury. We see a response within a few days or a week. We moved to a chronic degeneration model in the eye, which enables us to look at slowing down this phenomenon and look at that response within a month or so. And then we're really interested in looking at not only much slower neurodegeneration that occurs over multiple months, because of course, in humans, we know this neurodegeneration can occur over decades, but we were also really interested in just looking in other disease models that may enable us to study these functions in different contexts, particularly if this particular substate of cells is present in all these other diseases, as we saw in the human postmortem tissue. So what I'm going to do now is jump and actually have a look at a mouse model of amyotrophic lateral sclerosis, or Lou Gehrig's disease. So this is another piece of work that again was spearheaded by Kevin. It was also published last year, this time in Nature Communications. And what we've decided to use here is the SOD1 G93A high copy mutant mouse model of ALS. So SOD1 is superoxide dismutase. It was one of the earliest genes shown to have point mutations that were associated with increased risk of the disease. And there's some really beautiful papers from the mid-2000s by Serge Prydworski's lab at Columbia and Kevin Egan's lab up at Harvard that really digs deep into the underlying genetics of this particular mutation. And actually, this was one of the very first mutations that was shown to mediate the phenotype of this degeneration when it is expressed in glial cells and not in neurons, which was a big step forward for the field. So in this particular experiment, what Kevin has done is he's taken some sections of the spinal cord from wild type and mutant ALS mice, and he's done some in situ to look for this neurotoxic cell marker C3. And as we predict in the wild type mouse, there is actually none of these C3 positive neurotoxic astrocytes present. In the SOD1 mouse, there was a lot of these C3 positive astrocytes present. What Kevin then did was an experiment I told him not to do, and I'm forever glad that he did do it. But what he did is he took this mutant mouse, he crossed it with an astrocyte reporter mouse, the LDH101 EGFP mouse, and then he crossed that with all three global knockouts to remove the neurotoxic phenotype, again using this dirty mouse model. But what he ended up with was an I1 alpha knockout, TNF knockout, C1q knockout that harbored the SOD1 mutation and had an astrocyte reporter. And the idea here is that this particular mouse line would have the disease-associated mutation, but wouldn't have neurotoxic astrocytes. We see a decrease in the number of these C3 positive cells at the level of in situ. But the important thing is, do the mice actually perform better? Do they live longer? And indeed they do. So here we have a survival curve. In the vertical axis, we have the percentage of the mice alive. In the horizontal axis, we have the age of the mice in days. And you can see in the red line, the straight SOD1 mice are dying in within 3 to 4 months. And in the blue line here, when we knock out these factors, stop the formation of these neurotoxic astrocytes, we see an increase in the lifespan of these mice by about 50% or 2 months or so, which is a really large increase for this incredibly rapidly progressing disease model. And I should say here at this stage, we actually didn't see that there was a loss of central nervous system neurons here. There was no degeneration of the neurons, so to speak. But what we did see, there was still neuromuscular junction dysfunction. And there's a lot of talk in the field that ALS is actually an axonopathy and not a central nervous system degeneration disease, because we actually did still see this occur, and these mice still had some forelimb paralysis and were having difficulty eating, and so we euthanized them early, even though they had no degeneration centrally. So we could keep these mice alive, but the next question that Kevin asked was, well, is this due to a change in astrocytes or to a change in the other cells in the CNS? And so I'm just going to first talk about the fact that we were interested whether or not this would occur in the human patients, and then we're going to dig deep into the underlying probable functions of this. So the first example I have here is just actually now looking at some immunohistochemistry or immunofluorescence from human postmortem tissue. We have in a non-symptomatic or non-neurological control patient, we have obviously GFAP present, so we have astrocytes present, but we don't have these C3 positive neurotoxic cells. If we get patients that are harboring a mutation in the SOD1 gene, much like what we saw in the rodent, we can see these C3 positive cells present. What Kevin also did was looked in mutations in other ALS-associated genes like C9orf72, or even in patients that had no known disease etiology, so these sporadic patients. And when he quantified the presence of this C3 positive cells in either the spinal cord, the medulla, or in the motor cortex, across all of these different patient cohorts, you could see there was an increase compared to the non-neurological controls. And this is only a very small number of patients, half a dozen or so. So we then took advantage of some whole genome sequencing available from Project ALS. And he saw that across hundreds of different patients, ALS patients on the whole had a marked increase in the expression level of C3 compared to non-neurological controls. And again, when he passes those out according to mutations, either in SOD1, C9orf72, other associated ALS genes like FUS or TDP43, or again, these sporadic patients with no known etiology, he could again see that there was an increase in this C3 marker. So the next question that he had then was which one of these cells was actually changing. And so because he had this astrocyte reporter line crossed in, he could actually pull out the astrocytes or use Downs homogenization to purify the microglia. He could grow them in a dish and say, pre-symptomatically or just normally in the brain, do cells that are harboring this mutation have any transcriptomic differences or changes from the normal physiological state? And he showed that, lo and behold, there actually wasn't. We expected there to be more, but astrocytes that are either harboring mutation SOD1 or not, wild-type astrocytes the same, and so are microglia. And so the next set of experiments he did, which is one has been done by dozens, if not hundreds of labs around the world, was he gave these cells a pro-inflammatory stimulus, and he showed that when you compare quiescent and reactive astrocytes, there was a huge change, both up and down regulated transcripts in the astrocytes, and the same is true in the microglia. But the final piece of the puzzle was he took reactive astrocytes or cells that have been treated with some pro-inflammatory stimulus and compared the mutant versus wild-type cells. And when he did this for both astrocytes and microglia, he found that the microglia remarkably were similar regardless of whether they were wild-type or mutant, but the astrocytes that were harboring this mutation actually upregulated a whole host of different genes they didn't before, suggesting that their response to this stimulus was very, very different. And this is something that we're digging into now to try and understand the underlying physiology and the functional consequences of this. The other experiment that he did was to actually say, well, is the amount of stimulus that is required, is the threshold that must be breached to enter a reactive state also the same between wild type and mutant astrocytes? And so what we have here is just a normalized response of this reactivity, looking at the extent to which all the genes are upregulated against the stimulus input. And we can see at the maximum input, the wild type, the mutant cells are almost identical in their transcriptomic profile. But if we drop this stimulus back considerably, once we get down to 5% of the stimulus, in black here we have the wild type astrocytes. They're responding with about 50% efficiency, whereas the mutant astrocytes are actually responding close to normal control, with somewhere between 80 and 90% of the full reactive repertoire. And so we interpreted this to say that not only are the genes that they are inducing very, very different, but the ones that are similar across the wild type mutant astrocytes have to reach a lower threshold for this secondary reactive response to occur. And so maybe these patients that are harboring, or these astrocytes that are harboring these mutations, are just primed to become reactive in a way that the wild type cells are not. So I'm keeping an eye on time here, and Jorge hasn't yelled at me yet, so I'm going to keep going until he shakes his head and tells me not to. But essentially what we've talked about here is that microglia have the ability to induce this specific reactive substate in inflammation, but also in the context of different neurodegenerative diseases. We talked today about acute insult, chronic glaucoma, and also in this ALS model. And we know that these neuroinflammatory reactive astrocytes can release a neurotoxin. And as I said, I don't have more to say about that today, but we have identified this toxin and the enzymes that are required to produce it in astrocytes. And so we have some new conditional reactive astrocyte mice that no longer produce that toxin. We also know that if we block this neurotoxic reactive astrocyte formation, we can actually preserve not only the numbers of neurons that are present, we can also preserve their function. And so the question that really arises now is, how heterogeneous is this response, and what can we learn about understanding the specific substates of these cells that are occurring, and do specific substates occur across multiple different diseases? And I want to talk a little bit about this today because I think it's important in some of the conclusions I'll draw about human disease, but Philip Harsel in the lab is going to talk about this way more in depth with you in a couple of weeks' time. So the first thing I wanted to talk about was how seemingly simple but technologically complex this question was for us to address in the lab. And the first reason it was complex is that it's really, really hard to get astrocytes into single cell based gems, much like use like Dropseq or 10x technology. They seem to be one of the cells in the central nervous system that is largely excluded. And so just to show you how this looks, if we look at the very first Dropseq paper by Evan McCosco, we can see that even when you sequence around 700,000 cells in lots of different brain regions, the number of astrocytes that are collected is incredibly low, around 3 or 4%. And if we look at some particular brain regions like the cortex, we know that the percentage of astrocytes, when we just count the numbers of cells, is more closely aligned to 30 to 50%. And so there seems to be a dropout of astrocytes here. Then with the move to 10x, more commercially available, when we look in different brain regions like the nucleus accumbens, the hippocampus, the cortex, if we look at acute insults to morphine or more chronic models, such as the 5x FAD mouse model of Alzheimer's disease, we can again see that the numbers or the percentage of astrocyte capture is incredibly low. And this leads to incredibly low number of astrocytes across the entire study, and an incredibly low number of astrocytes from each individual mouse. And then once we start cutting this up into different substates and clustering, we're only ending up with a small number of cells or a dozen cells from each of these clusters. And we feel that this could lead to some artifacts of the interpretation of these data simply because they're underpowered. So what Philip did when he joined the lab was he actually worked out a way of increasing this capture rate. So he's now capturing in all the gems, 90% or so of them are astrocytes, which enables him to look at a large number of cells, but also give us huge numbers of cells from each individual animal, which is really important. So now when we cluster these cells out, we have different numbers of hundreds of different cells in each one of these clusters. So just to show you what he's done, he took exactly the same LDH mouse that Jennifer Zemanian originally took. He then captured a huge number of astrocytes, and in red here we have this huge astrocyte data set. In other color, we have our small non-astrocytes, as I said, so neurons and oligodendrocytes and microglia and all the other cells, but he's captured a large number of astrocytes. And so if we look here, we can see in this three-dimensional UMAP that Michael O'Day, a graduate student in the lab, has developed, has produced We have a huge number of these cells. Philip looked at them and the way that those responses change with response to inflammation and also with respect to different sexes. He knows that all of these cells are astrocytes because they express canonical astrocyte markers. And he also knows that there are substates of these reactive astrocytes or different clusters of these reactive astrocytes that are expressing very specific markers. And I'm sure he'll go into this a lot more depth. But what I did want to do is just talk about this one particular cluster here, cluster 8, which we affectionately in the lab call DUMAs, for no other reason than we thought it sounded silly. And these names don't really mean that much. And so we go for the Shakespearean approach, that is, a rose by any other name still smells as sweet. A reactive astrocyte subtype by any other name still has the same function. So IgTP positive astrocytes are called DUMAs. And so what are DUMAs? Well, they're a subset of reactive astrocytes largely present only in the inflamed brain. We rarely see them in the physiological brain. They are marked by interferon response genes like STAT1 and IgTP. Philip used some in situ hybridization to localize them within the brain, and he sees here with a co-labeling for IgTP to mark the DUMAs and SLC1A3 or GLAST to label astrocytes. They're largely present on the outer layer of the cortex, so layer 1 of the cortex just under the meninges, with processes extending down along these penetrating vessels. He knows it's actually not the vessels itself that are expressing this gene, because when he co-labels for the endothelial cell marker Claudin-5, he can see that the cell body has collapsed onto the vessel, but it's actually not being expressed by the vessel itself. He then also used some spatial transcriptomics to localize them more broadly throughout the brain. And we came across another problem. Of course, when you're looking at single genes with spatial transcriptomics, because of the low resolution, it's very hard to work out exactly where these transcripts are expressed, even though we can see there's a slight increase, but what does it mean? So Philip developed this module probing approach where he took every gene that was enriched in these DUMA subset of reactive astrocytes, and then he probes for them in the spatial transcriptomics as a module, and we can see then that these reactive astrocytes, or the transcriptomic profile of these reactive astrocytes, is localized to the outer surface of the brain, but also around the ventricles, both the lateral and the third, and tracking along this foramen between those two. And then Michael O'Day, who I mentioned before, was doing this three-dimensional imaging of these astrocytes, then took advantage of a different approach of investigating this. So on the left-hand side here, we have a normal single probe visualization following spatial transcriptomics, and we're using the 10x Visium system here. But what Michael took advantage of was this Bayesian next nearest neighbor analysis pipeline called BaseSpace, which was published by Edward Zhao recently in Nature Biotechnology. And what this does is it adds additional signal to adjacent spots with the same transcriptomic signature, or decreases the signal if they are different. And just by comparing these two single probe approaches, just looking at IgTP here, this DUMA marker, we can see with this increased or enhanced resolution, again, like Philip showed with in situ, we see that this DUMA marker is present around the ventricular system and not really largely elsewhere in the brain. So the big question arises here, and I'll rush through the last few slides in case there are any questions, is how did Philip find this subtype of reactive astrocytes when people haven't really described it previously? And I spent a lot of time, or at least a slide, talking about how the capture rate for astrocytes is very, very low compared to other CNS cells. And we think this is the reason. So here we have just a large data set of astrocytes, around 100,000 or so. We probed for this DUMA marker, IgTP. We can see that those cells cluster together. If we subsample from this around 10,000 cells, we can see that we are still able to cluster these cells together. But when we get down to around 1,000 cells, we can no longer see a cluster of these cells, even though there must be some cells present there. And we know that these DUMAs represent between about 1% and 2% of all the cells in the central nervous system in a reactive state. And if we just sort of harken back to this table that I showed you, if you need 10,000 astrocytes, we know that previous data sets, although they may be high quality on their own, they're simply not powered enough to cluster these cells together. So this brought up two important questions for us. Is one, has anyone actually described these unwittingly in the literature before? And two, is there a way we can take advantage of published data sets to see if these individual substates of cells are present? So I just want to talk about two quick diseases in my last few slides, if that's okay. no one's shaking their head, so I'm going to go ahead. So the first one is actually some work that was published 2 decades ago that actually showed the presence of this marker CXCL10 or IP10, which is actually another transcript that is, in addition to being upregulated in microglial immune cells, is also specifically upregulated in these Duma cells, these interferon-responsive reactive astrocytes. So here we have some nice immunohistochemistry looking at non-symptomatic patients and in Alzheimer's disease patients, and I'm sure you'll appreciate there is a large marked increase in the amount of CXCL10 staining both within astrocytes and also the numbers of these astrocytes. And so what Jessica Sadik, a postdoc in the lab, did then was took, in blue we have here Philip's large single-cell transcriptomic data set and integrated it with smaller published data sets that were using the 5x FAD mouse of model of Alzheimer's disease. So here we have this in purple, this data set from Naomi Habib. It was in Mikhail Schwartz's lab. In red and yellow, we have 2 different time points from a data set from Marco Colonna's lab. And needless to say, you can see that the data set that Philip generated is incredibly large in comparison. But if we actually look at what transcripts each one of these different clusters are expressing across these different data sets, I won't go into a great deal of depth about this dot plot here, but needless to say, at the bottom, we have all of these cluster 8 transcripts. On the vertical axis here, we have all of the individual clusters. All we're going to focus on is this cluster 8 or this Duma cluster. And if we actually just hone in on just a small number of these transcripts and look at these violin plots, which I think are easier to get our heads around, we can see that in the 7-month time point of this 5x FAD model from Marco Colonna's lab, we don't see the expression of this IGTP marker in control mice, but we see an increase in the 5x FAD mouse. And what's remarkable to us is these reactive astrocytes are present at the earlier time point at 7 months, but they're not present later stages of the disease model at 15 months, which leads us to hypothesize that these Duma cells are actually really important in the early protection or initiation of this degeneration. I'll comment that we actually didn't see them in this Habib data set because in addition to being extremely low powered, it actually didn't have incredibly high read depth as well. And so there simply was insufficient quality of the data for us to see these transcripts. And indeed, we can also check along other Duma-specific transcripts like IFIT-M3 and STAT1. We see the same story. So we then decided to look at other diseases as well, and some work out of Richard Ranzerhoff's lab over the past 2 decades has also shown CXCL10 positive astrocytes in both human patients with MS and also in the mouse demyelinating model EAE. And of interest to us was the fact that these CXCL10 positive astrocytes were also closely associated with blood vessels, similar to what Philip saw in his inflammation-induced model. And indeed, once again, when Jessica integrates these with the smaller data set here from Frank Quintana's lab, who we talked about before the talk started, we can see that with this really nice high-quality data set, we're just going to focus on the yellow here, which is the astrocyte purified data set. We can see again, we don't see the Dumas in the naive setting, but we do see them in the acute setting. It's actively demyelinating. And then in a final data set, Jessica integrated with this data set from Jonas Frissan's lab from the Karolinska. And unlike the other data sets that were unable to show Dumas in their own data sets, this particular data set was able to show a small number of Dumas. It was a slightly larger data set, and they were induced more in this stab wound. But we can see again, once we integrate it, we can robustly see that they're not present in the control setting, but they are present following this injury. So all of these data are free to everyone to go and search. We made these available before the publication even came out. So gliaseq.com was a website we set up. You can go and look at all of the bulk sequencing data that Philip generated, looking at the astrocytic response to inflammation with respect to sex and also with respect to time, which I didn't talk about today. We also have all of the single cell data, which we've collapsed into a bulk RNA sequencing, pseudo bulk RNA sequencing data set, so it makes it much easier to look at. We also did some non-amplification validation of this using the nanostring glial profiling panel, which actually contains a vast number of these markers, which is great. And we also produced an in vitro model to study their function. And so we have all of that sequencing data there showing exactly how these astrocytes respond to interferons. And then if this is not enough data for you, Philip and Jessica and Michael also set up a data set so you can actually go and look at each of the individual single cell data if you are so inclined. So you can see, for instance, like GFAP is upregulated in lots of different clusters, but also you can look at the spatial transcriptomic data and show GFAP while heterogeneously expressed at the baseline level, is upregulated across most brain regions. But you can also go hunting for cluster-specific markers, such as this one here, which is largely upregulated in deeper cortical and white matter tracts, really characterized by the expression of TIMP1. And you can see that it's not present in the non-inflamed brain, but is present in the inflamed brain. So the last thing I want to say as well, we've also been doing this in human samples. This hasn't been published yet, but it should be out shortly. But I just wanted to give you a little piece of pre-information about this. So similar to what Philip did in the mouse, Jessica has been doing in humans. She generated a large data set, again shown here in blue. She integrated it with other published data sets that were much smaller in scope and underpowered. So this one here from Liwei Tsai's lab and this one from a group in Australia. And you can see once you integrate them, there's actually some subtypes of these reactive astrocytes that were not described in the original paper, which we can now see by increasing the power of these analyses. And then, of course, the final thing that we're able to do is take our human data sets and our mouse data sets and integrate those together, which takes a lot of computational power and time. But I apologize, I've lost the labels here, but we can see that there are some subtypes that are maintained from the human into the mouse. And so here we have, for instance, neurotoxic astrocytes, and here we have these Duma astrocytes that are maintained across these two species. And so we think that these are really important models and subtypes of these cells for us to understand functionally, to try and get an understanding of what their roles may be in these diseases, rather than simply having an artifact of culture or an artifact of any particular mouse model. So that was sort of all I had to say, just to sort of rehash. You know, we know that some mouse models are very good at recapitulating astrocyte reactive states that we see in humans. I showed you some data from the Bead inclusion model of glaucoma and the SOD1 G93A high copy mutant mouse model of ALS, although of course there are others, and we mentioned the 5XFAD mouse and EAE, which are good models of other subtypes of reactive astrocytes. We also know that neurons must be susceptible to be killed by this astrocyte-secreted neurotoxin. The toxin itself is insufficient to kill them, so healthy cells are always safe. And we know that if we preserve neurons from this astrocyte-induced neurotoxicity, they actually maintain some electrophysiological function, which was a big surprise to us. And most importantly, we know that at least for the neurotoxic subtype of cells, this function and the transcriptomic profile is preserved from both rodents into human cells. And now we're investigating this in the context of these Duma cells and other reactive subtypes that Philip identified from his single cell studies. And then towards the end, I just showed you that these astrocytes do have this transcriptional and functional heterogeneity. You can go and look through the data sets for your particular favorite gene of interest, which is really important for trying to ascertain exactly which brain region you may be interested in studying if it's only reactive in one particular brain region. I didn't talk a lot about this today, but we'll also have, along with this human data, is that a lot of these AD-associated astrocytes, which seem to be specifically reactive only in Alzheimer's disease and not in age non-neurological controls, actually maintain similar spatial locations in both humans and also in the rodent inflamed brain, which we find really interesting. And we'll have coming very, very soon in a short number of months, we're just bug fixing our data set. But we'll also have searchable data sets for both the proteomic, lipidomic, and small metabolomic changes in these reactive astrocytes from a physiological to a neurotoxic state. And so with that, I thanked all of the people that did the work as we're going through, showing each of those slides. I have down here all the papers that I mentioned, both from Philip, Kevin, and Lily Babar. The data, as we mentioned in the last few slides, is all available, and I have enormous thanks to all of those supporters, both at the financial level listed here, but also to the families and the patients who have donated tissue and samples for us to conduct these studies. And with that, I would love to take any questions that you may have.
Okay, somebody. Okay, Alejandro Villarreal, go ahead.
Hi, Dr. Liddelow. Thanks for the talk. It was amazing, and thank you for sharing the data, making it available. So we definitely use it more than once. I have two questions. One maybe is more refers to the first part. And a second one related to the second part or the last part. To the first one is so you'll always look at the effect on neurons from these neurotoxic astrocytes. Did you? I know there are some works on this, but did you also look how these cytokines or other cytokines may act on microglia, like crosstalk, and maybe macrophages?
Such a good question, Alejandro. We have been very lazy. We have mostly looked at this axis from microglia inducing astrocytes and then astrocytes having an effect on neurons. And we haven't looked as much as what's happening in the opposite direction. So with all of this proteomic, lipidomic data and metabolite data that we've been generating now is the first foray that we're having into looking back at this now. We can look at exactly what molecules the astrocytes release and try and do those assays and studies and infer what the effect could be back on the microglia or other cells in the CNS. I will say that there's some beautiful studies out there done by other groups that have looked at this. And so I always like to use as an example the study by Julia TCW, who at the time was a postdoc in Alison Goet's lab. And she was using human iPSC-derived astrocytes with different ApoE expression. And she showed that the ApoE4 astrocytes have a decreased capacity to produce cholesterol. And of course, we know from Chris Boland's work that cholesterol is a really important trophic support molecule for microglia. In the brain, astrocytes produce practically all of the cholesterol. And so if you have less cholesterol, you're probably going to end up with an angry reactive microglia, which would then secrete cytokines that would activate the astrocytes. And you get this incredibly vicious cycle. Okay. And so I think I wish I had a better question, and I wish we had done all these, a better answer. We had done all these studies to tell you exactly what's going on, Alejandro, but we just haven't. Everyone in the lab has been really interested and focused on this one direction. But we'll put the data up.
It's already a lot of work, so no worries.
Yeah, but we'll put the data sets up soon, and I'm sure someone like yourself will look through it and notice something that is particularly interesting. We'll have the exact answer to what that means, and so we'll make them available incredibly shortly. Great question.
Thank you. And the second question is related to the clustering you did. Were you able to cluster these cells back to any pathway? And by pathway, I mean like transcription factors, or that could be determining, I don't know, 1 or 2 clusters or something.
Yeah, I guess the reason for wanting to know the transcription factors is trying to work out exactly how to induce individual substates as well. Correct? Yeah. So we've just started doing the transcription factor mining at the moment, actually looking at a sort of relatively new analysis pipeline and producing what are called bow plots. And so you can look at how individual clusters of these cells may turn on or turn off transcription factors at particular stages during their activation state. One of the things that we learned very quickly when we started doing this is all of the single cell that I showed you today is from a single time point. And so it's not particularly conducive to doing exactly that analysis because it's missing some really important changes that are occurring early. So we've actually gone back, and so Michael O'Day in the lab has been generating large single cell data sets from very early time points. And another graduate student in the lab, Paul Frizzell, is actually doing a similar sort of thing, looking at transcription factors required for astrocytic development as well. So we should hopefully have that answer really soon for you, because it's something that we immediately realized was important, but then didn't have the data set to look at. Okay. And when we pulled the available published data sets, they had this problem of just being underpowered again. And so we were essentially getting spurious results out of them. So hopefully shortly.
Yeah, sure. Thank you. Thanks for the questions. The answers, sorry.
Well, thank you for the questions. And I'm sorry I didn't have the perfect answers, but they're things that we think about all the time.
I just wanted your opinion and whether you look at it or not. It's super perfect. Thanks.
Great.
Yeah, thank you, Shane. Thank you for the really nice talk and for putting all the information together. And also thank you, as Alejandro said, for sharing this information on the web, because sometimes many times we in all the not completely developed world look at that to do research, investigating on the things that you already did that we can't do. So thank you very much for that. And my question is gonna be on neurons. Are the neurons doing something for the astrocyte to read them and to kill them? I mean, are they not normal and astrocytes are sensing that? are they expressing something, or are they not expressing something?
Yeah.
Like I remember the don't eat me signals of the neurons of microglia, and I say, okay, maybe they are stopping the expression of something on the membrane, and so astrocytes are reading that. But do you have any clues about that, or any answer?
Yeah, this is oh, Javier, this is a great question. This is something we have agonized over for years now. And so even when I was back at Stanford, we did some very early experiments that showed that different motor neuron populations have different susceptibility to this toxin. So we showed that large caliber alpha motor neurons are particularly susceptible, but smaller caliber preganglionic and gammas are not susceptible. And so one of the initial things that we did was just actually some RNA sequencing of susceptible versus non-susceptible neurons to look for a receptor, right? Is there something that's on these cell surfaces that is mediating this response? And we never found anything. So the sort of devil's advocate or alternate sort of interpretation of that is we just didn't prepare the study very carefully and we missed the interesting biology. The way we interpreted it at the time was, well, there's nothing that's receptor mediated that's driving this. Since that time, we've actually investigated this a lot further to try and determine exactly what's going on. Injury, like I said, seems to be a very powerful mediator of this susceptibility. And so one of the things that we played with for a while was maybe the injury actually just puts holes in the cells and the toxin can just get in. And so normally there are no holes in a neuron, the membrane is very tight. And so could this be something that's mediating the toxicity? And so we did this artificially by using, say, like DPC to punch holes in cells. And that could actually drive susceptibility of neurons that were otherwise non-susceptible. So we sort of thought that that could possibly be the mechanism for a long time. But we now, of course, there's a lot of literature showing that different mutations in neurons, in genes expressed by neurons, can mount some susceptibility. And so that doesn't really fit with the just hole in the membrane hypothesis. What we do know is, though, that the molecules induce a caspase-3 mediated apoptosis. So it's a very well-described pathway, seems to involve PERK ATF3. We've mapped the pathway out, but we still have a black box between the cell surface and the initiation of that pathway at the endoplasmic reticulum, or that cell stress pathway at the ER. And so I don't have a better answer than that. I'm really sorry, but we know how the toxin's being packaged up. We know how it's being delivered. We just don't know what happens between the cell surface and the initiation of the pathway, but we know it gets into the cell. So maybe it's random endocytosis. Watch this space. We're going to spend probably way more years digging into this deeper and deeper.
Thank you.
Patricia.
Good morning, everyone. Can you hear me? Yeah.
Yep.
Well, I'm Patricia Casina from Uruguay. I want to thank your talk because it's a wonderful talk. I admire your work from many, many years. And I would like to ask you whether you have found a model in which the reactive astrocytes are induced for other kind of stimulus besides the triple cytokines, interleukin-1 alpha, TNF alpha, and C1q. Because all your work goes that microglia induce for this triple expression of cytokines, reactive astrocytes. But I was wondering whether you have found in some other model the reactive astrocyte induced for another form, perhaps some metabolic stimulus or some perhaps stimulus of the Toll-4 receptors, or I don't know. I want to.
Really, really great question, Patricia. So
because you have shown beautifully that the expression at the transcriptomic of the astrocyte in different conditions are quite different. So that was a very important observation. So I wonder whether you. Yeah.
No, this is a really, really great question. I think this is something that the field on the whole is struggling with right now. Is we have all of this transcriptomic data, but it almost means naught if we can't actually then infer that functionally. And so we've spent a lot of time trying to work out exactly what the mediators are that produce each of these subsets at the transcriptomic level. So the original neurotoxic cells, which is the TNF, IL-1, and C1q, when we actually aggregate all of these different clusters together and just pseudo bulk them all and say, what is the transcriptomic profile that comes out? that is the one that seems to take over, just because the degree to which those genes are upregulated is so high that it quenches all the other smaller subsets. So by doing the single cell and by powering the single cell properly, you can get enough numbers of these cells. And the DUMA signature, the interferon responsive signature I talked about today, actually does require TNF, I1, and C1q, which we think is coming from the central nervous system, but it also requires interferon. And without interferon, you can't produce those subtype of reactive astrocytes. And if you only use interferons, you also can't produce them. So we think that what this is you need C1q, IL-1, and TNF from the microglia in the central nervous system, and then you need interferons from the peripheral immune cells. And because they're located either on these penetrating vessels or around the ventricular system, their location is very strategic, that they can actually respond to both the peripheral and the central nervous system signals together. And so that's a subset that is a combination with the C1q, but also requires other things. The other substates of the reactive astrocytes, this thrombus 1 and 4 reactive substate, or all the others, there were 10 of them there that I didn't talk about, Philip will talk about much more, they require molecules that are not TNF and I1 and C1q. They require something else. And they have extremely subtle transcriptomic changes, smaller numbers of genes being altered, both up and down regulated, and to a lesser degree than what we saw these neurotoxic cells being upregulated. So they just get lost in this bulk sequencing. So I find that incredibly fascinating. And so we're hot on the heels of a border or a scar producing reactive astrocyte. So we think that we know exactly what the molecules are that induce that. It's not TNF, it's not I1, it's not C1, it's nothing that we would have normally thought about. But of course, we've done it in a dish. We don't know if it's real, and so we're trying to validate it in vivo at the moment. So Rachel Kim, one of the graduate students, is what she's validating at the moment. But there is an almost infinite number of these substates, I think, to go looking after. I talked about DUMAs. We have fast-acting DUMAs, which we call Zoomers. We have slow-acting DUMAs, which we call Boomers. Are they the same cell type that is just changing its gene expression with time, and they need the same initiators? Or are they occurring at different times and they require different initiators as well? That's a really fascinating question that we're trying to dig into. So that was a very whirlwind answer. Sometimes,
no,
yes,
but it's okay. The rationality is important then. So right. you may have the same stimulus, but according to the place you are looking at, perhaps you have different response. That's
exactly. And there's a lot of people working now on just spatial heterogeneity at the physiological level. So Omar Bayraktar, who was in David Rowich's lab, talks about different cortical layers. Baljit Kark's lab talk about astrocytes in different brain regions having different transcriptomic profiles as well. And that's definitely true at the baseline level. And some of their transcriptomic changes are different in each of those brain regions, even when following the same initial insult. So you can have an inflammatory astrocyte in the cortex that may be different in the striatum. We haven't seen that as much with these very large data sets. The baseline heterogeneity remains, but the reactivity responses that are laid on top seem to be very similar from region to region, although the expression levels may be slightly different. What we don't know is if there are any functional differences there. And that, I think, is the bit that's going to really matter. Because if there's no functional difference, they're the same subtype of reactive astrocytes. But they could be transcriptomically similar, but because of the local environment and the other cells that they're interacting with, they could actually have very different functions. A lot more neurons or neuron cell bodies in the hippocampus than there are in the cortex. The astrocyte's going to be interacting with a lot more neurons. There's probably going to be a lot more neurotransmitters around. When we study it in a dish, we miss all of this, right? And so I think their biology is incredibly complex, and that's why we have to keep jumping back and forth between in vitro and in vivo as much as possible, because it's very easy to overinterpret these results.
Okay, thank you for the opportunity to hear you. Okay.
No, it's a pleasure. And that, like I said, great question. Something that we think about a lot, and that's why we like to make these data sets sort of available as early as possible, because we look for stuff that we understand, and all it takes is someone with a slightly different level or area of expertise to find something way more interesting than we can find in these data sets. So we'll keep putting them up there as soon as we get them.
Thank you.
Okay, Jane, I have a couple of questions also. Thanks for the talk, and it's amazing, the whole project that you have and results, and hope that you will kind of push the field forward. I remember that I have heard some researchers from Harvard working with these IPS cells, kind of differentiated them into astrocytes in a model of ALS from patients with ALS, and they found that some prostaglandins were involved in the killing of the neurons. So, do you see something like this in your cells, in the neurotoxin cells?
Yeah, this is a really interesting thing. So this gets back to that original diagram that I had, where most of the time cell biologists look at normal physiology and look at the response to acute insult. And once you change altered pathology or altered physiology, I think this is where things get really, really interesting. So we at the moment haven't actually looked at the delivery or the secretion of neurotoxins from astrocytes, let's say with altered physiology or chronic pathology. It's something that we are doing, but we are doing it in a way where we're not starting with astrocytes from the mouse models. We're starting with embryonic stem cells, and we're engineering them to have specific point mutations associated with disease, where we sort of know what the functional outcomes might be, or at least the pathways involved in those particular gene products. So it's something that we are slowly building up to, but we just had to do all the groundwork to get those engineering approaches on board. I will say, though, that the other time this becomes really interesting is when the genetic mutation changes susceptibility of the neurons. And so Eric Huang had a really lovely paper out, or Eric Huang's group had a really lovely paper out in Nature earlier this year, showing that mutations in progranulin or deletion of progranulin in neurons can actually increase susceptibility to complement components coming from microglia, particularly important in frontal temporal dementia. And the wild type neurons are not susceptible to these complements proteins, but the progranulin knockout neurons are. And so again, everything changes once you look at the altered physiology or the chronic pathology on both sides of that equation. So we have focused mostly on normal physiology responding to secondary insult, because we had really good models for that. And now we're slowly branching out to start investigating how that particular well-defined pathway changes in the context of these diseases, much like we did with the ALS stuff. We're just now doing it with a lot higher fidelity. Yeah, great question, though.
Maybe another question. In some of the multiple sclerosis models, you see some inflammation in the meninges, and then you have these IGTP astrocytes, like in this molecular layer of the cortex. So could they be involved in the process of multiple sclerosis? Yeah,
Yoni Kipnis asked me this the other week, actually, because of course he's really interested in the meningeal macrophages and how they may be interacting. I would guess yes, absolutely. The location that they have really up against the meningeal vessels, or just the first sort of branch of these penetrating vessels, and like I said, around the ventricular system, I think they're definitely interacting with those specifically located immune cells in those regions. We have done nothing in that space. I'm waiting for someone like Yoni to have a really beautiful data set of those meningeal macrophages so we can do some receptor ligand interaction sort of analysis and see, is there something that they could be secreting that the astrocytes could be responding to in addition to this interferon? I think it would make all the sense in the world to me. And particularly because we know from Frank Quintana's beautiful EAE astrocyte single cell that they are there, right? So when you have this sort of systemic inflammation that mediates this degeneration of the myelin, we know that these substates are produced. We've only just started to sort of scratch the surface there a little bit. So the question we've also been thinking of is there a possible insult, or is there a disease where that location would change? So is there something you could do where it's normally layer 1 cortex, but now all of a sudden these cells are present in the hippocampus? What would mediate that change? And so maybe it's just when you have some vascular breakdown. So maybe having these transient ischemic events that occurs, or maybe when you've got sort of amyloidosis in the vascular wall, which occurs during AD, or maybe when you have a big stroke, you're going to have these astrocytes occurring in different regions because it's actually the interaction with the outside world, so to speak, that's causing this change. Oh man, there's so many cool things that you could investigate in this space. And we can't do. I'd love to do them all, but I'd have to hire way too many people. I like to keep things small, but so many cool things to look at. Yeah.
Another comment could be that if these gap junctions and the formation of networks in between all of these astrocytes could influence like of the spreading of phenotype in astrocytes, or how are involved, if they are involved, this astrocyte network.
Yeah, that's a really exciting point. We actually, for the longest time, I thought that we had a supplemental figure in our original Nature paper from 2017 that talked about uncoupling of astrocytes. And it turns out I didn't. But one of the things that we found was, at least for the neurotoxic phenotype, I don't know about any other subtype of reactive astrocytes, but it's something that I think should be looked into. For the neurotoxic phenotype, they seem to uncouple from their neighbors. So you don't get these beautiful propagating calcium waves, whatever those calcium waves mean, but you don't get them propagating across large regions. Now, we did this in vitro. The reason it never went into the paper originally was we could never validate it in vivo. We were not an in vivo calcium imaging lab. And so we didn't make it in because we didn't feel confident in it. But a few people have asked this recently, and there's been a couple of papers out like this work from Dave Colkin's lab. Melissa Cooper was the first author earlier this year, showing that you can actually have connexin 43 or gap junction breakdown in the context of insult and injury. So we've been interested by this, and we were also interested by the fact that when you make a neurotoxic reactive astrocyte, it can't replicate, it doesn't divide and make more, and it doesn't secrete cytokines, the same cytokines, to make more of itself. So it requires an external source, microglia or some peripheral immune cell, to release the cytokines to initiate that change from physiological to reactive. And we also saw in addition that the astrocytes have no cell autonomous ability to turn themselves off once they enter that reactive state. So we think that the master regulator, the mafia mob boss, so to speak, is the microglia or the immune cell comes in, gives a signal to respond, moves away and does other things because they are motile. Astrocytes are not motile. But at some stage, the pathology or the infection has been mediated, and an additional set of microglia or immune cells comes in and releases different cytokines that then turn that astrocyte off again. And that I find to be a really intriguing mechanism, because if you remove the microglia, we know the response doesn't occur. What happens if you have mutations in microglia that mounts a greater response, or doesn't bring in the turn off signal response? And how does that play into our ability to treat patients who may have slow or fast progressing disease or early onset, et cetera, et cetera. So the interplay between these astrocytes and all the other cells on the upstream and downstream side is something that I think we should all be looking at, because it's incredibly fascinating.
And also in the relationship in between the different subgroups of microglia and oligodendrocyte precursor cells and your neurotoxic astrocytes, because they may change altogether.
100%. Yeah, we like I said, it's the dirty little secret of cell biologists. We often don't look at altered physiology or chronic pathology, and that I think is something that we have to start moving towards, because that could change everything that we know. It's likely that it's just altering it slightly, but those alterations are going to be really important for us to know, because otherwise we're going to be giving patients future therapies that actually don't work on those patients at all. It's something that I constantly think about.
Great. Thank you, Shane.
If there are no more questions, may I ask
of course,
for an opinion? So Shane, again, I need your opinion. We've been discussing for a long time with the students, if we are talking about gain of function, pro-inflammatory gain of function, or on the other side, loss of homeostatic functions.
Mm-hmm.
But are the astrocytes, our concept is like astrocytes are all the same, basically, initially, and then gain or they lose function. And are all the same? Are they preset for going into pro-inflammatory role because they are originally different? What do you think, or if you have any experimental data or anything, what do you think about that? Are they preset to achieve this, to gain of function, the pro-inflammatory gain of function, because they are like dormant or the sleeping immune cells
Yeah.
that are engaged in that program, or are all basically the same and they are in the wrong place in the wrong time?
Yeah, that's a big question, Javier. That's a good question. That's a really big question. You know, are astrocytes different in different brain regions? I guess is the first part of the question. I mean, yeah, I mean, they're transcriptomically different. But the question is, are they inherently transcriptomically different, or are they just transcriptomically different because of the environment they're in? And so I would love someone to correct me if this does exist, but I'm not sure that anyone has done a transplantation study where they have taken, say, hippocampal astrocyte and put in the cortex, or cortical astrocyte and put in, say, striatum, and seen if they have different functions. I guess the best example is maybe the human to mouse transplants that like Steve Goldman had done previously, where he took human astrocyte precursors and put them into mouse, and they grew into what appeared to be human-sized astrocytes. But we don't know if that's because they were put into a different region, or if that was just a species difference, because we know human astrocytes are much bigger, right? The sort of same to same experiment, I don't know. Maybe someone has done this with organoids now. I know like Sergio Pasca does these connectoids now, where he takes like striatal cortical astrocytes and organoids and puts them together. Maybe he could switch the cells. I will say though that, you know, astrocytes, if we think just about synaptogenesis, we know that spark is really important for synapse formation. It is expressed by a very specific, spatially restricted subtypes of reactive astrocytes, same as thrombospondin and things like this. So that at least suggests that there are some differences in known genes, or genes that are associated with known functions, but we don't know if that's because of where they are. You know, if we look at other things like the number of synapses that astrocytes are wrapping around, you know, the calculation from the like Baljit-Kark's lab or from Charlotte Eroglu's lab are somewhere between like 50 to 100,000 synapses in different brain regions in the mouse, 1 to 2 million in the cortex of the human. But I mean, would an astrocyte just wrap those neurons that are near it, not that they have this inherent capacity to grow bigger or grow smaller? I don't know that we have that answer yet. And I wish I could tell you what happens if you pull out different brain region astrocytes and put them in a dish and they maintain the transcriptomic profile. We just haven't seen that yet, which suggests that they're just responding to the environment, which is not to say that heterogeneity is not there. I just don't know that it's inherent, if that makes sense. But if someone has proven that, I would love to know, because I've missed it. I think that would be really fascinating, that you have just astrocytes that are primed and developed for exactly that brain region, and they wouldn't work anywhere else. And if you had some flipping of those regions, it could lead to some astrocytopathy that would be a disease that we don't really know about. That would be fascinating, really fascinating. I'm sorry, I don't know the answers to a lot of questions, but I muse about lots of things.
Thank you very much.
Yeah, I've been thinking about that one for a while. I'll get back to you if I think of something or I notice something.
Okay.
Yeah,
that would be amazing. It's a discussion that we always engage with the students, because it's really interesting if we can translate astrocyte from one side to the other. Not only regions, but roles. It's amazing.
Right, exactly. Yeah, it's a really intriguing little thought problem. And like I said, if anyone else on the call knows as well, I would love to know, because I don't know. I think someone should do that experiment.
Yeah.
It's a really cool experiment, and it would teach us so much about just baseline biology of the cells. It would be really, really exciting to know that.
Yeah. Okay. Thank you once again. Thank you.
Thank you.
Hey, Jane. Okay. Thanks a lot, Jane. So no more questions. So it was a wonderful talk, and also for being with us all this time. It was a long talk.
No, absolute pleasure. I apologize, I went a little long.
No, not your presentation, the questions.
No, please, the questions are the best bit. So if anyone thinks of things offline, please feel free to email me, or if I don't reply to email, tweet at me, so I publicly am shamed if I don't respond. But this was an absolute pleasure. Thank you very much for the invite. It was great. Discussion was awesome.
So hope to be also with you next year, maybe, or
That'd be fantastic. In
Yeah, yeah.
person would be really lovely.
Yeah, yeah.
Take care, everyone.